All right, so we started off with DNA, and we took that DNA and we made mRNA through transcription. And now we need to take that mRNA and make a protein. And we're going to do that with a process called translation. So we are going to translate the mRNA into proteins. Now, this is going to take the help of something called rRNA, which is ribosomal RNA. Which makes up ribosomes. Remember, way back in the day, we said that ribosomes were created proteins. That was their function in the cell. Well, rRNA is what makes up these proteins. So rRNA makes up proteins. And tRNA is called transfer RNA. It brings amino acids, which we know that amino acids are the building blocks of proteins. We know that from way back. Brings amino acids to mRNA to create a protein. All right, so we're going to have 3 types of RNA in this situation. We got our mRNA that we made. We got our rRNA, which is going to be basically a ribosome. And then we have our tRNA, which is going to bring our amino acids to the party. All right, so let's talk about, we have to talk about translation in 2 different ways. We need to talk about actually how translation occurs, and then we need to talk about how the mRNA is read and creates a amino acid chain. So I'm going to start with that first. So let's say, remember, we had DNA was all green. So let's say our DNA code that we had was ATGCUA. I don't know, CCG. All right, we're just going to have this. We're going to make it short. When we translate that into mRNA, okay, A has to go with U, remember, because there's no T. T goes with A, G goes with C, C goes with G, U, there is no U in DNA. Oh my gosh, silly. We got to move this to a T. Oops. Silly. All right. T goes with A goes with U, G, C. All right. Now, notice I have these grouped in groups of 3 because this is how they're going to be read. These groups of 3 are called codons. Groups of 3 nucleotides. All right. So now we have our new tRNA that has to have, tRNA is going to come along, and tRNA, I should have drawn it for you. It's shaped a little bit like a clover leaf, like this. And at the bottom, this is where it has its code And at the top, this is where it holds on to its amino acid. All right? So this code at the bottom is called an anticodon. And it's essentially just the opposite of the mRNA. So U has to go with A goes with U, C goes with G, G goes with C, A goes with U, U goes with A, C, G. So now these are the tRNA anticodon. Now, each one of these anticodons that are at the bottom here codes for a specific amino acid. So how do we know which is which? We have a little chart, okay? So this is where the chart comes in. Nope, move over there. Move. All right, so you will, I promise you on the EOC, you will have to do one of these. And you will have to use this chart if I can ever get it to get bigger. All right. Let me scoot it up. All right. So this is the first letter of the anticodon right here. And then this is the second letter. So if we're looking at this code, so now we need to call it, this is the amino acid, AA. All right. AUG. So I go to here, A. Then I find the column U, and then I find one that's AUG. Here we go. And it's MET, which is methanone. But we just put MET. Then the next one, we go to C, then U. So we're looking in this box right here, CU, and then find the one CUA. That's leucine. All right, next, we go to the next one. This is CCG. So we go to C, then we got to go to the second box, which is C, and then we find the CCG, and that's proline. All right? So this is our amino acid code, and these are all going to be linked together by a peptide bond, which we will discuss later. All right? I will promise you, you will see this on the EOC. Now, they could, instead of using this box one, they could have one that looks like a circle, but it works in the same way. So the circle one works in the same way. You would start like with this first one, A, then pick the next one, U, then find the G, and then you have methanone. Okay, so the circle works in the same way. I think the circle one is actually easier. I don't know which one that they'll put on your test. But I promise you're going to see something where you have to go back and forth between a DNA code, mRNA, tRNA, and an amino acid. Okay, so you have to remember that A always goes with T in DNA, but A goes with U in RNA, and that will pretty much help you figure this all out. So that's how that's on the letter end. So that's the smaller narrowed in picture. So now we really need to talk about the sort of big picture, how this occurs. All right. So we know that the mRNA is going to be divided or sort of, they're not physically separated, but they are read in chunks of 3. Okay. So they're not physically separated by anything, but they're read in chunks of 3. All right. So let's scroll down here just a little bit. And I have this picture, which is a very, very detailed of what I'm going to describe to you. So it's a little more detailed than I'm going to require of you, but this should help you understand what I'm saying. All right, remember, just like in translation, we had 3 steps: initiation, elongation, and termination. We're going to have the exact same 3 steps here. So initiation, number 1, this is where you have the rRNA or the ribosomal complex is going to attach to the mRNA and brings the tRNA to the mRNA. Now, how does it know where to start? That is a good question. So you have this big long mRNA, but we know it has a poly A tail and a 5' cap. And so how does it know where to start? Okay, there is such thing as a start codon. So mRNA has a start codon, which is, AUG. This is a start codon. So tRNA is going to bring with it the anticodon, which is UAC. There is only one start codon or anticodon, codes for methanone. All right, and that is the first start codon. So on the mRNA, as you can see in this picture, you'll have AUG, which will bond with UAC. And it's carrying the amino acid methanone. And this is the first one to come to the party. So when the ribosome goes down the mRNA and sees AUG, it will stop, it will attach, and the tRNA will come. Okay? So this is Now, the next step, elongation. The tRNA is going to continue to bring amino acids, which I'm abbreviating AA, to the mRNA. And the amino acids bond to each other with a peptide bond. All right, so here in this picture, you can see this is just going to be, the ribosomes are going to be moving down. I'm going to scoot this up. So here's our mRNA, all right, and they're going to be read in groups of 3, okay, and the ribosome complex is like over a lot of them, and then you'll just have separate tRNAs. They're hard to draw, I'm not going to lie, carrying these amino acids. And eventually these amino acids are going to be bonded together like this, and you'll have, and this is just, the ribosome is going to move down the mRNA and continue to add them until you just have blocks of amino acids all attached to each other. You have a long chain. And they're just going to be a whole bunch of amino acids all chained together. Because once it drops off its amino acid, this tRNA leaves and it'll go pick up another amino acid. Okay? So what you end up with is this really long chain of growing amino acids. Now, just like we started this whole thing with a start codon, we're going to end it with a termination codon. So we have termination, and this we use a stop codon, unlike the start codon where there's only one, there's actually 3 different stop codons. Okay? So there's 3 different ones. But either way, they will get here, and once this once the tRNA hits a stop codon, it will just release the amino acid chain out into the world. And we call this, in the end, what we have created is a polypeptide. Poly means many. Peptide is the type of bond. So we've created a long string of amino acids. Now, I guess technically we have now created a protein because proteins are formed from amino acids. But we're a long way from anything that can function as an actual protein. Okay? So once we have finished with translation, we then have to turn this polypeptide into something that we can use. Let me find my picture. It's around here somewhere. Ah, there it is. Then we have to turn this protein into something we can use. So that is where protein formation comes in, protein folding. So each amino acid has a specific chemical compound. So what we have created here, let me just make this a little bigger. What we have created here, this is a primary protein structure. It's just a chain of polypeptide, or a polypeptide chain, all right? That's a primary structure. Now, what will happen is each one of these amino acids has some kind of charge usually. Well, some have charges, some don't have charges, all right? But they won't be able to stay into a chain for long. They're going to fold in on each other with hydrogen bonds. So hydrogen bonds are going to create either a helix or a sheet with all these amino acids. And you have to remember that a protein is made up of thousands of these. I've got 5 written here. But an actual protein will have thousands of these amino acids. And they'll all be attracted to each other in different ways for different reasons. And that creates a situation where these proteins are going to fold into a sheet or a helix. And we call this the secondary structure. Now, we have a helix or a sheet, and so a helix just basically looks like this, and a sheet is going to be like a folded sheet of paper, and I really don't know how to show you what that looks like. A folded sheet of paper. I don't know. Something like that. Anyway, these two are going to be attracted to each other, and so when you have a helix and a sheet interacting, we call that tertiary structure. Helix and sheets form bonds. So at this point, you have at least two polypeptide chains, all right? You took 2 different polypeptides. One became a helix, one became a sheet. Then they interacted with each other. This is going to be your tertiary structure. Then when you take a whole bunch of tertiary structures, 2 or more tertiary structures equals a quaternary structure. So tertiary means 3, quaternary means 4. Now, why do I tell you all of this? First of all, I tell you all of this to tell you that translation only makes a polypeptide chain, not an actual functioning protein. A functioning protein is created once the amino acids all interact with each other to form different shapes, all right? The important and the biggest thing that you should get out of all of this is a protein shape determines protein function. And the shape is determined by the combination of amino acids. So, you should be, at this point, you should be asking yourself this question. What could go wrong? Now, if a protein's shape determines its function, so every protein must have an exact shape in order to perform its function. That shape is determined by the chain of amino acids that we created in transcription. Where could this go wrong? Well, considering that amino acid chain came from mRNA, which came from DNA, there's 1,000 places where it could go wrong. All right? So we could have a mistake in the DNA itself. So when we copied it the first time, we made a mistake. We could have a mistake in the mRNA. So when we copied the RNA to the mRNA, we made a mistake. If we have a mistake in either one of those two things, then we are going to get the wrong amino acid sequence. And by the way, these mistakes, are called mutations. And every mutation, so if we make a mistake in DNA, remember we said that when we create the mRNA, we take out introns and leave the exons. If the mistake we made happened to be in an intron, then we're not going to have any problems. It's fine because we cut it out and it's no different. It makes no difference. But if that mistake is in an exon, that becomes a real big problem because now we're going to end up with the wrong amino acid sequence, which means we're going to have the wrong protein shape, which means we're going to have either no or the wrong protein function. All right. So the mutation can be as small as in DNA, let's say, the mistake could be, it's supposed to be AUGC, but somehow, why do I keep saying that U's are in DNA? Duh. ATCG, let's say, is supposed to be the code. But when we replicated our DNA, we have ACG. So we substituted this one thing. All right. That when that goes down to the mRNA, oh my goodness, so sorry. I must have done that with my thumb. The mRNA sequence, so here's mRNA, is going to be UUGC, and then our anticodon is going to be AAC, because remember our codon's only 3. AAC amino acid is, I'm just going to use the chart, AAC aspirinine. Okay. Now let's take this the original. So originally it was supposed to be ATC. So if I do mRNA from ATC, then I should get UAG. And if I do the tRNA, I'm just working backwards. tRNA, I get AUC. And AUC stands for isoleucine. So just by changing, all I did, guys, was change this one letter in this one sequence. And so instead of isolene, which is what it was supposed to be, I ended up with asparagine. And because they're different amino acids, they're going to interconnect in a different way, and they are going to create the wrong protein shape. Okay, so my main point in showing you this is that if we make a mistake, either in DNA replication or in mRNA, then we can end up with the wrong amino acids. And so probably on your EOC, they are going to have one where they try to get you to tell them, okay, this is the right DNA sequence, this is the wrong DNA sequence. What amino acid is going to be wrong? And so you have to go through the whole chain to figure it out. Okay? So that is basically how we make a protein. It took us 3 days, but we did it. We made a protein, and then we learned how they can be messed up if we make one little mistake.